So, our lecture this evening is Neurophysiology in Action from Dr. Oli Steele. Dr. Steele is a lecturer in physiology at Brighton and Sussex Medical School who teaches across phase one of the undergraduate medical programme, whilst also managing a small research programme interested in the way in which cells in the brain communicate and how this goes wrong in diseases. So, without further ado, I'll hand over to you, Oli.
Thank you very much, Zach. That's a lovely introduction. I'll just share my screen, and if Zach, you could tell me that you can see that. That should be working there. Can you see that, Zach?
Yes, that's all good.
Wonderful. Excellent. Cool. Let's make a start then. Yeah, as Zach said, my name is Ollie, and I'm a lecturer at Brighton and Sussex Medical School. And it's a real pleasure to be talking to you today, this evening rather, about neurophysiology in action, something I'm particularly passionate about. So before we do begin properly, I use a software called Slido in my teaching. And it's a fantastic tool to sort of get you involved in the teaching as well. So if you would be able to scan this with a smartphone or join at slido.com with that code there, that would be fantastic. Then you'll be able to answer some of the questions that I have prepared for you, some of the activities as well, and you'll be able to ask me questions towards the end as well. So I'll give that, let's say, give that another 10, 15 seconds or so just for you to join. Don't panic if you miss this now. Whenever a question pops up, this will be in the bottom left-hand corner, so you can join then as well. And I think Zach might also be putting this in the chat as well, so you can join whenever. Cool. I think that's about 10 seconds, so I'll have some coffee to let you carry on, and then I'll start. Wonderful. So the first one's really easy. It's not a trick question at all. I'm just genuinely really interested in where you're all watching from. We've got UK, England, there's a big representation there. Hi, Wickham. Saudi Arabia as well. That's considerably more exotic than my home office in the south of England. Southeast England, yeah. Okay, Southampton. So I've got some friends over in Southampton at the moment. That might even be them who logged in. God, Peterborough, Crawley, Peacehaven. Peacehaven are some real local towards Brighton as well, then. Some excellent representation from Ukraine as well, and from the kitchen. So thank you for being very precise with that one. Okay, that's lovely. Thank you. That's really interesting to see where you're all watching in from. So before I start with the content of this and sort of talk about neuroscience, I thought it worth sort of just briefly introducing myself so that I'm not some random person just talking at you over the phone. I see Paris as well. That's awesome. So my name, again, Ollie, I'm a lecturer in physiology at BSMS, and I'm really interested in how these brain cells communicate with each other. When I'm not doing that, I'm teaching across years 1 and 2 of the medical program at BSMS, and then when I'm not doing either of those two, I'm usually out chasing my dog, Frodo. I can't give a talk these days without a photo of Frodo being included at some point. So here is said obligatory photo of Frodo. I hope you appreciate that. So I did my GCSEs and A-levels in Angmering, Angmering sixth form, before going on to Exeter University, which is where I did my medical sciences degree. That was my BSc in Exeter. Loved it in Exeter, really nice there, but then moved to Cardiff, where I started my master's degree. At the same sort of time, I also worked as a research associate for a little while, and got involved in some really cool projects that I'll talk to you about towards the end of this presentation. After Cardiff, then headed back down towards Brighton. So I've not gone far. I've been around a little bit and then come back to where I am. So it's really nice for me and my family are here. And in Brighton, I did my PhD in neuroscience and took up the lecturer position that I currently hold. So sort of not nearly as exotic as Paris or Saudi Arabia, but traveled around the south of the UK a little bit. What my job kind of entails at the moment, my wife refers to me as the man of many hats because I seem to have a lot of different responsibilities. A lot of that is teaching centric, education and teaching centric. I act as a deputy for the head of physiology teaching at BSMS. I lead a certain module, so reproduction and endocrinology, as well as doing some programming and coding as well. And I tried to work out what a day in the life was when I was preparing this presentation. And it's really quite hard because no day is really the same. But sometimes I'm lecturing to students often 200 students at a time, and sometimes it's smaller group teaching. But with the role comes the ability to do some things that I'm really passionate about, so things like outreach and widening participation, and speaking to people like you. So yeah, I really, really enjoy doing that. At the same time, as Zach mentioned, I do manage a small research program. I thought I'd give you a little overview of what that research is now, and I'll talk about it a bit more later. But if you know what it is now, then maybe you can ask some questions if you had it. As we said, I'm really interested in how neurons communicate, and in particular, the protein ApoE. ApoE4 is the one I'm really interested in. It's a risk factor for Alzheimer's disease, but you might have even seen it in the news at the moment because the actor that plays Thor, I think it's Chris Hemsworth, tested his genes and found that he's got ApoE4, and so has this hugely increased risk of Alzheimer's disease. So it's one that we should all be aware of, and my research centers around it. So bear that in the back of your mind for now, and we'll talk about that a bit more later. Cool. So what I wanted to do today, first off, I really wanted to dispel some common neuroscience myths. I think we've all probably heard them at different points. We've all just seen them around. So we're going to play a game of Neuro Mythbusters. You're going to help me out with this. We're then going to go into sort of a mini lecture, right? I'm going to sort of teach you in the way that I teach at the medical school here, so you get a bit of an experience of that, exploring neurons as a functional unit, before then using that neuroscience knowledge to control muscles. I then sort of, obviously, whilst that's really cool, I then try and talk about the clinical applications of some of this stuff before talking about neuroscience research, so particularly the stuff that I'm working on. But this one at the bottom, I hope you can see my pointer, this one at the bottom, number 6, I really need you to appreciate how cool neuroscience is. And if at the end of this you don't, then frankly, I've not done my job well enough. Neuroscience is really, really impressive, and I really hope I can sort of get some of that across today. Cool. So, section 1, Neuro Mythbusters. I don't know if any of you will have watched Mythbusters. I'm increasingly being told that it's quite an old program now, which is dating me, but it was one of my favorite programs as a child. Essentially, they'd be given a myth every episode, and then they would bust it together. So we're going to play a neuro-specific version of that. I've got a series of questions for you. So that first myth or fact is the left side of the brain is more logical and analytical, whilst the right side of the brain is more creative. If you could answer whether that's a fact or a myth, give it a couple of seconds longer. We've got, oh, it's nearly 50-50. Oh. I just watched this all evening. And I'll give you another couple of seconds and then we can crack on. Looking like fact is starting to win, 55-45. Okie dokie. Okay. Yeah, and the more I mention the word fact, it's going up higher and higher. So this one, I suspect you will have heard of, this idea that one side of the brain is much more involved in creative and arts and that sort of thing, whereas the other does the math and the logic and the engineering or whatever it is. This one is a total myth. This one is quite hard to pin down where this one came from. And whilst it is true that our brain, if you look at it, has 2 hemispheres, you see on that there that you've got 2 halves to the brain. That is true. There is a physical halving of the brain. But the fact that one side is particularly responsible for the creative side is kind of nonsense, right? Now, it might be true that there are certain areas that are particularly involved in, say, speech, like Wernicke's area. If you have damage to the Wernicke's area, which is on one half of the brain, then you have severe impediments in speech. But that's a very specific case. And normally, really, whenever we're doing something particularly creative or logical, our whole brain is going to be being activated, right, rather than one side over the other. A lot of this probably came from a famous chap in neuroscience called Mr. Gage. Mr. Gage ended up getting a metal pole through part of his brain, destroying part of the brain. Somehow he survived, which was quite a marvel. But he had deficits that were specific to that bit of the brain that was gone. And then I think it kind of grew from there. So yeah, number 1, that one is indeed a myth. So let's go on to the next one. We only use 10% of our brain. This one has been popularized in films like Limitless or Lucy. Is this a myth or is this a fact? I seem a lot more confident this time that it's myth. Um, give you a couple of seconds, get over 50 people. Okay, yeah, looking like we're still favoring myth at this point. So again, like I said, films like Limitless, films like Lucy. If you've seen either of these films, Limitless, Bradley Cooper takes a pill, accesses 100% of his brain, and becomes a genius, kind of overnight. Lucy, on the other hand, is considerably weirder. I think as Lucy, played by Scarlett Johansson, gains more and more control over her brain, she begins to be able to control space and time, and very strange film. But this idea that if we access more of our brain, we can do more. Correct. This one is indeed a myth as well. This one can actually be pinned down to a misquote of a neuroscientist in the 1800s that suggested we have hidden potential, right? We're only accessing 10% of our potential. But then a journalist misquoted that as potential and being part of our brain. We're only using 10% of our brain, and then it's kind of just grown from there. So really, this one's not true. There aren't hidden parts of our brain that we can activate, become super smart, or to help with your exams. I do apologize. And indeed, in some cases, whilst we're not using all of our brain all the time, there are certain conditions where more of the brain can be activated than is healthy. And this might be a condition such as epilepsy, where the brain becomes dysregulated and hyperactive. So yeah, this one is indeed a myth. So thank you and well done on that one. Your brain has roughly the same number of neurons in it as the Milky Way has stars. Is this one a fact or a myth? Looking very confident on this one, 85%. Okay, yeah, I think we are pretty much decided that this one is a fact. This one is fantastically a fact. There are approximately, and the number really differs. It's really quite hard to count this many neurons, but there's approximately 100 billion neurons in the mature human brain. So that's about the same number of stars as a small galaxy inside each one of our heads, which is absolutely fascinating. Try and put that into sort of another term. A piece of tissue, a piece of brain tissue, the size of a single grain of sand that you might find on the beach, would have approximately 100,000 neurons. So these things are really, really small, but ultimately control all of our being. It is quite hard to count, as I said, and some of these kind of calculations and guesstimates end up coming from liquefying brains and trying to count them. And lots of the problems are really that different parts of the brain have more neurons than other neurons. So this one is true that there are a bafflingly large number, but the exact number is difficult to say. Well done on that one. This is the last myth, or fact rather. Our brains generate electricity, which controls our bodies. 100%. Wow. Give you just a bit longer. You're looking quite confident on this one, though, that this one may well be a fact. Give you a second or so longer. Okay, I think we've pretty much decided as a group that this one is indeed a fact. Myth is making a late charge. No, not quite enough. So much like Thor, we do generate electricity. Cells in our brain do indeed generate electrical activity. Obviously not quite to the same amount or level of control that Thor had, but we do indeed generate electrical signals, and these pass throughout our entire bodies to control our bodies. So what I wanted to do, well done, thank you for doing that really well for me. I think I only caught you out on one of them, so well done. What I want to do is to dive into a mini lecture here, sort of in the style of the teaching that I do at the medical school. There'll be some questions at the end for you to check your knowledge and understanding of the things we've covered. And really, this really is in the style of the teaching I do at the medical school. So hopefully you get a flavour of that. Cool. So neurons are the functional unit of the brain. And it's important to think about these as cells. Like any other cell, they have a cell body or a soma. You'll have the nucleus here, protein synthesis, everything else you've been learning in biology, that all happens in there. But then they have two quite striking features. They have dendrites that stick out the top, and then they have a long projecting axon. So they are quite distinctive in their shape compared to a lot of other cells, but they have a functional role as well, rather than just looking different. These neurons use electrical and chemical signaling to communicate between each others to both respond to inputs, perform computation, i.e. make a decision, and then produce an output signal. So remember that they respond to an input, they make a decision, and then they produce an output signal. So as I said, you have these different features. You have dendrites, you have a soma, and you have an axon that projects all the way down here. So your signal will be carried out towards your synapses. So these little sort of feet at the end of each of these axon projections, these axon terminals, these are where synapses happen. Synapses are connections between neurons. They're a physical gap for a chemical signal to pass across to in turn generate an electrical signal. So all that stuff you'll be learning in chemistry about sodium and potassium moving across, different amounts of sodium and potassium will move across a membrane at a certain point, and that will build up a charge, the same as the charge that we have in our batteries, or this battery here, for example. It's just a charge, a current, and a voltage. So if you remember this textbook image of a neuron, and I'm sure you'll have seen plenty of textbook images of cells and other parts of biology, but in reality, no part of biology is anywhere near as neat as it is in a textbook. So I want to show you some images and videos of real neurons, and hopefully you recognize some of these features. So these are both taken by colleagues of mine. This orange gold looking one by a colleague of mine in Southampton, Dr. Warming, has grown neurons on a glass slip and then essentially dyed them yellow to sort of highlight the contrast so you can get a better view of them. And you might just be able to notice these hot spots. Some of them appear white because they're so bright, but these sort of circles and spheres, they're those somas, those cell bodies you just identified on that image prior. And you can see each of these projections, these sort of finger-like projections coming off of them. This will be the axon coming down towards the other neurons' dendrites. So you can recognize the features, but it obviously looks a bit different. What you then have here in this green image, taken by Professor Coyer here at Sussex, actually, is neurons communicating with each other. These have a dye that when some of those ions move across the membrane to induce an electrical current, they glow green. So you can get a proxy of electrical activity from looking at these. And now, I'm biased, but I'd say that looks not too dissimilar to stars twinkling in the night sky, which is why I quite like that myth from before. So this is neuronal communication happening in real time. If we go from these sort of cellular models a bit higher up, and we look at an MRI, so this image on the left is actually my head, that's my ear, and as we go through the skull, you can see the blood vessels on the outside of my skull, that's the bone, and then here you start to see that sort of classical wrinkled structure of the brain. The wrinkling increases the surface area to allow for blood and oxygen to get to it. This little region is called the cerebellum, deals with your movement. That's my eyes, are about halfway through, just going past it. Here you come up towards the corpus callosum, some fantastic connective tissue, and that's my spinal cord running all the way down my neck. We then head towards the other side of the brain and it kind of replicates. So I'll pause that. But if you look at it at that level, it looks like a big grey mess, and you'd be correct in saying so. But a fantastic study by, I can't remember what country they're from, but it's called the Clarity Study. What they ended up doing was they made a bit of the brain completely see-through and then got the neurons to glow green. So I'll try and play this. I do apologize if it deafens you.
Into the center of a mouse's brain, into the hippocampus where memories are formed. Into the center.
You don't need to get the video. I want to show you the structure of the brains. And what you can see is if we're looking at the hippocampus, which is this bright green layer in the middle, the hippocampus is the bit that deals with learning and memory. But you can start to see some of those little hotspots like we saw on the previous image. Those are those somas, those are the neurons, and they're projecting up and out here, up out towards the cortex, which is where all our decision-making is occurring. So the brain is beautifully structured as well, but it's not just how it looks, it's kind of also how it functions. And that's what I'm most interested in. So one really important thing to try and understand with neurons is this idea of neuronal integration. And neuronal integration is this idea of these neurons have to make a decision, and they have to make a decision based on the inputs they're receiving from beforehand. So as in pretty much all of my teaching now, I have to make sure there is a Homer Simpson GIF. You'll be surprised at the variability of the Homer Simpson GIFs. You can broadly split your neurons into excitatory or inhibitory. If it's excitatory, it will encourage further activity. If it's inhibitory, it will prevent further activity, okay? So in this scenario, say we have a gray neuron here that hasn't made its mind up, it doesn't know what it's going to do. And a green neuron, an excitatory neuron, signals onto it. I'm sure you can imagine, you end up getting electrical activity. That's one of those spikes that we see. That is electrical activity in real time. Very simple. If we then have a gray neuron that hasn't decided and it gets an input from an inhibitory neuron, well, we then don't get any electrical activity, right? And this is really, really simple, but this is also how it works. In this scenario, however, you've got more excitatory signaling than inhibitory signaling, and the neuron has to make a decision and weigh up the number of different inputs that are coming onto this neuron. Now, in this case, you'd likely have electrical activity, but this is a very, very simple scenario. Obviously, in reality, you would have a whole lot more neurons. Now, if anyone can remember the number of neurons, it's about 100 billion neurons. Now, each neuron is suggested to have about 7,000 synapses on them. The maths there adds up to about 700 trillion synapses. So these neurons are weighing up 7,000 inputs and deciding whether or not to fire electrical activity. And all of our brains are doing this in such an ordered fashion that we're able to have this conversation as we speak now. And that's indeed what you're seeing there in that green video there. They're doing this in a beautifully ordered fashion. That is neuronal integration. But often it doesn't always go up to the central nervous system, up to that galaxy inside our heads. Sometimes we have to skip that pathway, and these are called nervous reflexes. A nervous reflex often can come about due to noxious or indeed painful stimuli. Imagine you touch something hot. Before you realize you've done it, you've pulled your hand away because it's hot. There's an evolutionary advantage to doing so. We need to protect ourselves. The way it does this is that an afferent signal will head up towards the spinal cord. And then instead of going up to the brain, it will then pass back down towards the muscles. You can see these efferent signals. So it heads up towards the spinal cord, is afferent, and away is efferent. It brings about an effect. This in turn leads to muscle contraction and indeed protective movement. So sometimes we can skip that galaxy inside our heads for our own benefit. Excellent. So I now have those questions just to check your understanding. This is how I do it in my lectures, prepare the students for the end of year exams. So at which point of the neuron does electrochemical communication occur? Soma, dendrites, nucleus, axon, or synapse? Give you a couple of seconds longer. Okay, we're up to about 50 again. That's looking good. Okay, let's dive into the answer. Yeah, spot on. That is indeed the synapse. Well done. So dendrites is an interesting one. The dendrites, you'll often have synaptic communication onto them, but it'll be happening through the synapse. It really is that synapse that's the important bit. And it could be a dendrite connecting to an axon or an axon connecting to a dendrite. The other answers are unfortunately just kind of more incorrect, but the synapse is the correct answer. What I'll also do after I do these sort of questions, I'll then show the students where that was in the teaching material so they can help with their revision. Okay, this one, which direction do efferent nerves transmit signals? Remember, this is efferent. Is it towards the brain or away from the brain? Don't worry, I get this wrong all the time. It's one letter different. It's a very cruel question. Up to about 50 already. That's really good. Correct. It is away from the brain. Remember, efferent is effect. So spot on. Well done. those 10% who got towards the brain, I'm in that 10%. I always get that one wrong. So well done to those who got that correct. Oh, only 9% now. You can't change your answer now, you know it's true. Cool. So thank you very much for that. Again, there's the answer to that. And that was sort of an overview of some of the style of teaching that I tend to do at medical school. These neurons are considered functional units of the nervous system, and they communicate via electrochemical communication at synapses. So what I want to do now is I want to try and I've taught you the theory of it, and I think by and large you probably all believe me that there is indeed electrical communication in our bodies. But what I'm going to try and do now is try and visualize this in real time. Now, this requires a little bit of technology that in the past has sometimes broken. Zach and I did test it before this, and touch wood, this will work again. So it's a bit of kit made by a company called Backyard Brains. Backyard Brains, anyone can buy this stuff. I'm in no way affiliated by them. I'm not paid to say any of this stuff, although the amount I talk about them, perhaps I should. And this allows us to see electrical activity in real time. You may have noticed on the back of my hand, I've got a little sticker there. It's got a little metal button on the back. There's no needles or anything like it. It's just a sticker if you've ever had an ECG or an EMG. I also have two just here as well on the muscle in my forearm. What I'm going to do is I'm going to connect a couple of wires to the back of these pads and hopefully be able to visualize that electrical activity in my body. So if I can change my screen, hopefully you can now see this green screen here. If I press start, cool. So that's wiggling about and not a lot is happening. What that screen is looking at is the electrical activity that's happening in my arm at the moment. There's not a lot. And if I move my fingers with my other hand, you might get a little bit, nothing major. If I move my fingers about, you start to see a little bit of activity because that's my muscles moving them. Or more precisely, that's my brain sending an electrical signal down my spinal cord and down a motor neuron in my arm to control those muscles. And if I really tense my arm, you see a lot more activity, okay? So I'm hoping I'm kind of proving in real time that it kind of exists. Now, if I had a willing volunteer, if I was doing this in person, I'd be able to do this a little bit differently. And I tried to convince my wife to do it, but she wasn't willing to be electrocuted in the name of science, which is fair enough, I suspect. I have something called a TENS machine here. Some of you may have used the TENS machine. I definitely used it a lot in physiotherapy. What this does is it sends a signal that contracts the muscles. So you can see as I'm putting these pads on my other arm, you can see the electrical signals coming through from this arm. What this will do is it will bypass my nervous system. It will send a signal from this arm down these wires and into these pads here, and it will activate the muscles in this arm. Okay. So if I turn that up a bit, okay. So if I just wiggle my fingers, you can start to see a bit of a signal, but not a major one. It needs to be a big one to get over the threshold because you've got to activate muscles. So if I tense that, if you look at this hand over here, can you see what's happening to those muscles when there's a spike of electricity? That's not me doing it. It's much more convincing when I'm doing this on somebody else, but I can't do that over Zoom. So what's happening is, again, I'm bypassing my brain and I'm hacking a nervous system to do so. Those signals don't look as ordered. You might have noticed correctly that this hand was sort of twitching a lot more than this one, which was quite controlled. And that's really because it's not coming down direct nerve pathways, but just broadly activating lots of neurons in the right sort of area to cause movement. So it's never going to look quite as controlled. So that's hopefully convince you that indeed we do have electrical activity running through our bodies and controlling muscles. And indeed, if you have the right bit of kit, you can actually control somebody else's muscles as well, willingly or otherwise. Cool. So, yes, very cool, very exciting. But, you know, why would a medic care about something like that? You know, when might this be relevant? So I have a question here, and I'm really interested to see what you guys come up with. What clinical scenarios might this bioelectricity be relevant? So think about whether you've seen it in medical documentaries, dramas, Holby City, scrubs, ER, whatever it is, when this bioelectricity is there. Either you're looking for it, or it's there when it shouldn't be, or you can use it to treat things. Really interested to see what you're coming up with. So paralysis is coming up as the big one at the moment. That's a new one. I've not actually had that one when I've asked this question before. It could be, but there's potentially lots of different causes of paralysis. And for example, if it's damage to the spinal cord, then it could well be, but it's the absence of any electrical activity because they're not going down the spinal cord. ECG is a really good one there. Epilepsy diagnosis. Oh, these are fantastic. Yeah. So seizures, spot on, right? That's when you've got too much electrical activity going on as well. Testing nerve damage, stroke, neurological diagnosis, physical therapy. Yeah. TENS, menstrual cramps as well. Yeah. So that TENS was basically what I was just doing with these pads here on my arm. So, this is awesome. Spot on. This all looks really, really good. Resuscitation as well. Yeah, like a defibrillator, for example. That is again using bioelectricity. It's everywhere and controls everything. As much as I talk about it all the time, it is actually there. It is important. Cool. Spot on. This is really, really exciting. Let me show you them. I've come up with a couple. I tried to group them into main areas. One of the most obvious ones is kind of what we were just doing, which is EMG recordings. So electromyography, which is just recording the electrical activity from muscles. Now, I'll let you in on a secret. Scientists are really not a creative bunch, and we tend to name things in the most boring way possible. So if you break that word down, electrical activity, myo, Latin for muscle, graphy, picture, right? So this is an electric muscle picture. If you ever come up against anything that you don't really know what it is in science, look at what they've called it and break the word down. You'll get pretty close. Some of these conditions you might want to do EMG testing for might be in things like diabetic neuropathy, traumatic nerve damage, indeed paralysis, or wasting conditions such as amyotrophic lateral sclerosis and others like that. So EMG is pretty much what we were just doing. And I do have a slight aside because we've talked about some of you mentioned about ECGs, which is your heart. They produce electrical activity, but so does your brain. Only two types of tissue that does. But other tissues will respond to it. So you've seen muscles, but it's not just the muscles in your arms. It can also be the muscles in your gastrointestinal tract. Now, this is an admittedly not fantastic diagram, but what it shows you, this tube is your gastrointestinal tract. And as you go down from the pharynx down to the esophageal sphincter, so your stomach is just below here, this graph on the right is showing a spike in muscle tension, or indeed as a result of electrical activity. As you can see, as you go down this pathway, they become ever so slightly later from each other. So they're tensing almost rhythmically. So top tense, it relaxes, then it tenses, and it relaxes and pushes it down. This is peristalsis, right? So it's muscular contractions happening as a result of electrical activity throughout our body. So even helping in things like digestion as well. Visualizing brain activity, several of you pointed that one out as well. So these would be EEGs, again, electric, encephalo, another word for head, and graphy, picture, encephalography, or EEG because it's a real mouthful. What's really cool about when you're measuring the whole brain's activity all at once is that you see these quite apparent waves, these waves of electrical activity that wash over the brain. And some of these waves are more common in certain states. So, for example, delta waves, particularly common during sleep, I believe. Where you might be interested in using this clinically, you can use it for the diagnosis, as some other people said in the chat, about things like epilepsy, looking at sleep disorders, but interestingly also brain tumors. A brain tumor is not ordered, organized neuronal tissue. It's aberrant cell growth that doesn't display normal activity. And as a result, you can see a pocket where you don't have that electrical activity. So you can maybe get an indication of a brain tumor. Obviously, wouldn't be the only way you do it. You might look at a scan as well, but it can help. So that epilepsy one, the one top there, and again, several people mentioned it, that there is a healthy EEG recording. This is an individual who was experiencing an epileptic seizure during a EEG recording, and you can very quickly see that that activity becomes completely out of control and completely aberrant. Wonderful. So visualizing heartbeats, as we sort of alluded to, the only other part of the body that produces electrical activity is indeed the heart. So if you're an electrophysiologist, you're interested in electrical activity and how that controls physiology, and you're either really interested in it in the brain or you're interested in it in the heart. These are the two main spaces where it creates its own electrical activity. There is also this the gut brain as well, but that's slightly different. So the heart generating its own electrical activity, you often see these on these ECGs. If anyone's had to have an ECG, they'll know what this feels like. It's just those sticky pads that I've got on my arm at the moment. You might have them across your chest and down your leg and arms. And you can see abnormalities in heart rhythm this way. So things like tachy and bradycardia, where it goes too fast or too slow, or atrial and ventricular fibrillation, where different components, valves in the heart open and close a bit too regularly. Fantastic. So that's where there is some clinical use. There's loads of others that we could have gone into using advanced prosthetics. The possibilities are endless. Bioelectricity is everywhere. It's the basis of all life. I'm very biased in that statement, but I will stand by it. So what I wanted to talk to you about is that I've shown you how it can be seen in the lab and how we can do it on willing volunteers and show there. But if we're actually in our research environment in a lab, how do we get access to this electrical activity, and how do we do it in a meaningful way, which is something that I was quite keen to wander through. So there's a huge breadth of neuroscience research, and I'm just going to talk about stuff that I do really, because it's easier for me to talk about, and I wouldn't want to do anyone else's a disservice. But having a look at, say, Sussex neuroscience, for example, or indeed any of the other neuroscientists across the world, there's a huge breadth of it. So I particularly use a technique called wholesale patch clamp electrophysiology. It's quite a mouthful, but what that is putting a needle onto a neuron and visualizing its activity in real time. So on this image on the left, you see this is my microscope. Underneath the microscope, just here, I have some brain tissue. That's a slice of brain tissue with some neurons in it. And you might see these little glass filaments coming in from either side. These are electrodes. Now, if you come to this gray image on the right, this gray image is of a neuron. You might recognize that soma that we saw before. You might recognize the dendrites at the top and an axon projecting out the bottom. But this sort of smudge coming off the right is one of those glass needles over here. So the needle has to be smaller than the size of a single neuron. And there's, 100,000 of them inside a single grain of sand. So this needle is beyond tiny. It has to get in contact with the surface, rips a very small hole in the surface, and then allows the solutions to mix. Once you've done this, you can use some really cool technology to then visualize that activity. And if I go here, that activity can look a little bit like this. So these are action potentials. If you've ever seen an action potential in a textbook, this is where they come from, right? So we are visualizing that electrical activity in real time from a single cell. So it's a really powerful technique. Some of the research that I've done before coming to BSMS in Cardiff, I was really fortunate to be involved in a program looking at stem cell derived neurons to try and model childhood epilepsy, so CDKL5 deficiency. There was a lot of cell culturing here, differentiation, and electrophysiology. So this is where I first started playing around with those needles that I talked about. I stayed in Cardiff, worked as a research associate, and we did some work on stem cell transplantation therapy in Huntington's disease. We needed to, in Huntington's disease, what you have is part of the brain is essentially degraded, and one idea to try and treat Huntington's disease is to replace that lost matter with stem cells. Now, for this to work, what needs to happen is those stem cells that turn into neurons, they need to functionally communicate with the rest of the brain. And as I'm hoping you're appreciating from some of the talks that I've given today about how complex that system is, that is a very tricky thing to do. So my job on that program was really to try and understand whether and how these neurons were communicating with the rest of the brain. I then moved to Sussex to start my PhD. Here I got really interested in apolipoprotein E, as I said, the major risk factor for Alzheimer's disease. And we tried to understand how this gene is changing the way neurons communicate. It doesn't always have to be in a particular disease, because even the findings that aren't in a disease are still applicable to diseases. You need to understand the basic biology before you can treat a disease, if that makes some sense. One project that I'm working on at BSMS at the moment is whether or not we can use ketamine to try and restore some of this functionality. So we saw these neurons, and I showed you those action potentials a minute ago. They fire lovely action potentials and they look really healthy. But when you put ApoE4 in it, which is the Alzheimer's variant of the gene, what you can see is a marked decrease in the firing of action potentials. These Alzheimer's, if you want to call it that, neurons are less active. It's obviously a stretch to push it to all of Alzheimer's disease, but ApoE4 is the major risk factor for Alzheimer's disease. So in our model, these ApoE4 neurons were less active than ApoE3 neurons. We then apply ketamine, however, we can restore some of that functionality. And some of this work has been done by some fantastic students working with me at the moment. So Bradley and Nico are currently in the lab doing some experiments, and Lewis is actually in Scotland at the moment doing some amazing work up there. So really, really fortunate to be working with some amazing people. And that's one of the joys of kind of working in science is the number of people you work with. Additionally, myself and a colleague, Alex, worked with some fantastic colleagues to try and produce a mammoth review trying to explain what on earth ApoE4 does. It's very confusing and it does almost everything. So if anyone particularly wants some rather dry bedtime reading, then that paper may well tickle your fancy. So I've talked for long enough, so I wanted to A, not bore you with any more of my talking, but B, just run through those things that I said we try and do at the beginning and see whether we've achieved. First, we wanted to try and dispel some common neuroscience myths, and I think you did fantastically at that, playing Neuro Mythbusters. Hopefully you can use some of that information with some of your family and tell your teachers at school and stuff. We then did a mini lecture where we explored neurons as a functional unit, and then use this new knowledge to control someone else's arm. I've got and robot claws there because in theory you could use this to control little motors as well, couldn't you? We then understood some of the clinical applications of neurophysiology. It's not just me thinking it's really cool and mucking about with it. There is a real clinical use case for it as well, used by doctors in practice. I then lastly showed you some research at Sussex, and I sincerely hope that I have, at least in part, tried to show how cool neuroscience really is. And as I said before, if I haven't done that, then I think I've done my job badly. So nothing else for me to say apart from thank you for your time. I sincerely appreciate it. And I'm going to flick on to the next slide, which should bring up some of the questions if you have asked any questions in the Q&A function. Indeed, you haven't. You're welcome to put your questions there. If not, then Zach and I can try and answer some of the ones in the chat if there are any.
Yes, yeah, thanks, Ollie. We have had a couple of questions come in on the Q&A. But yeah, folks, if you have any questions at all, please do pop them in either in the Q&A or you can just ask them on the Slido. We have just under 15 minutes, so we'll try and get through as many as possible before we finish up for this evening. So we've got a couple here, so we'll start with those, Ollie, and then we'll move on to any more that pop up. Does wearables for health monitoring use bioelectricity to measure? Is our first one.
That's a great question. It depends on the wearable you're talking about, right? I mean, some of the really fancy ones have things like pulse oximeters in there that can tell your blood oxygen levels. And that's not really bioelectricity because that's passing a sort of wavelength of light through and seeing how much comes back. And I don't think a lot of the ones that use look at your heart rate actually use the bioelectricity. Again, it's disruption of light. but I could well be wrong. There may be a few out there that do. But I wouldn't be surprised if there are, and it becomes increasingly more common in time because it's becoming more and more accessible. Like I said, anyone can buy this backyard brains kit that I've got out on my desk here. So yeah, I wouldn't be surprised.
One of the other ones that we've got here actually is almost linked to one of the ones that you have here on the Slido. So, how did you go into researching after university? And I suppose that links into this question: why did you choose research over a more practical element of neuroscience?
Yeah, that's a great question. It's this sort of this one at the top as well as the one that's in the chat, right? How did I get into research? I got into research because I like the detail. I enjoy the problem solving on a really fine level, and I enjoy the being in an area where we don't know what the answer is, right? I mean, research is both exciting and terrifying at the same time because you come across your textbook and where your textbook kind of finishes, that's just what we know at the moment. And researchers kind of get to that back page and go, yeah, but how does that work? What would happen if we did this, right? And it's working in that kind of gray area that I was just always attracted to. I enjoyed the challenge of it, if that makes some sense. So I guess that's probably how I kind of got into it. I did a research based undergraduate. My master's was research, my PhD was research as well. So if you're interested in it, following tracks that way definitely help. Over a more practical element of neuroscience, I'm guessing you're probably talking about things like neurosurgery and so on. To do that, really, you need to do a medical degree as well. And if I'm honest, there are more questions that I have not answered yet in the sort of understanding of science than I'm willing to leave alone yet. So I'll keep doing it for a little while. But it takes on a really there's a real benefit for it, right? Those neurosurgeons who are doing those neurosurgery rely on the scientists who've done the science in the first case. So it really is all linked together. So I'll answer that one, I think. I hope that answered that one. Okay. Do you want me to crack on with these now, Zach? Or...
Yeah. If you want to do, we've had quite a few come through on the Slido. So yeah, if you want to jump
lovely.
onto it, to a few of those.
I can jump on that one. That's a nice, easy one. So EEG helping the diagnosis of a brain tumor. Realistically, it probably isn't one of the first ones you do. You'd probably end up doing a scan or something that way. It would be a bit more obvious and conclusive that way. An EEG detects the amount of electrical activity that's coming from your brain. And when your brain is as a functioning unit, you see waves rippling across your brain. A brain tumor is not normal neuronal tissue and therefore doesn't fire those normal neuronal signals. So if you were to pass an EEG over an area that had a brain tumor, you would see non-normal signaling, which is kind of how it could give a clue that it's there. But like I say, realistically, you probably wouldn't start with this. You might have done this and gone, oh, that's weird. There's a patch over here that doesn't. And then you'd refer them for scanning as well afterwards. So it was a bit of a tidbit there, but I hope I didn't confuse anyone with that one. Let's try and answer that one. I tried to sort of talk about that one at the beginning. That I cannot answer that one in any more detail other than it's massively varied. I don't do this every evening, and this is part of my working day. So it's, for me, a massive plus. I wouldn't want a job where I was doing the same thing every day. But that can be a lot of teaching on some times of the year during term time. Outside of term time, there'll be a lot more research-based stuff. I might be in the lab, or I might be writing a paper, or whatever, right? So really, there is no typical day, and that's part of the joy of it for me. So I hope that helps. Let's go to this one here. In your opinion, what is the most interesting brain disease, disorder, or surgery, and why? Come on, I'm biased. I'm going to say the one that I research. I'm really interested in any form of neurodegeneration, probably principally, how the brain degrades and how the signaling in particular is interfered with in those pathways. So yeah, anything like that. But I think for me, I'm probably an odd one. I don't really have a disease or disorder that I'll focus on forever. I'm much more interested in the technique and the principle of electrical communication. And wherever that ends up taking me is wherever I'll end up going, right? Which is why I did some stuff on childhood epilepsy, for example. it's about that electrical signaling and how that goes wrong for me. So yeah, in my opinion, it's got to be neurodegeneration, mostly because I research it, but also because it's becoming the leading killer around the world at the moment, and is such a prevalent condition. Yet we have such a poor understanding of what's really happening and how we can treat it. So yeah, that would be the one for me. But they're all interesting because I'm a neuroscientist and I find them all interesting. Why did I choose biomedicine? I'll fly through these like as fast as I can to like rattle through as many as we can, and then Zach, you can just throw something at me when we don't have time. Why did I choose biomedicine? I chose biomedicine because I was particularly interested in the science more than the clinical side of it. So you may have caught on, I'm not a clinical doctor. I am a doctor of philosophy, a doctor of science, right? That's what a PhD stands for. So I'm really interested in the science and how that can help those who are doing the clinical work do their job as well. So I chose biomedicine because I knew I wanted to follow a science research-based way. So that's why I chose biomedicine. I hope that helps. Favorite uni out of the ones you've been to? I will say that Brighton and Sussex Medical School is the favorite one I've been to. Although I wasn't a student at BSMS, that's tricky for me to answer. It's really tricky. I had really great times at all of them, right? But I like Sussex. I like Sussex a lot, and I like where it is in the world. It's one of my biggest things when choosing a university was to be somewhere you're happy. So yeah, for me, probably the Sussex way. What is it like to work in a lab in terms of general experience and your job role? How much independence do you get? Oh, that's a great question. What's it like to work in a lab? Working in a lab is, again, very varied. Some days you'll be doing experiments. Some days you'll be doing calculations just to work out how to do your experiments. Some days you'll be writing papers. Some days you'll be at conferences. So it's very varied again, but it's more hands-on than it is sort of managing a team of people or doing teaching or anything like that. So it's a bit different. In terms of job role, again, depends whether you're sort of a junior scientist in that lab and you're doing the experiments, or you're a more senior, like a postdoc, or leading the lab. Then you might take on more sort of admin responsibilities for the lab rather than just being, on the ground doing experiments day to day. So how much independence you get, that varies depending on the lab you're in, how big the lab it is, but also how much independence you like, right? Some people don't like it and want to work in a team, so find a route that works for you there. But often you can be very independent. If I've got a cool idea in my head and I want to know if this works, there's not a lot stopping me doing it apart from financially and ethically and things like this. So independently, yeah, you get quite a lot there. What careers can you go from here? Oh God, I thought I had the adult job now, and I didn't think I was going to look for careers past it. Careers go from here, right? I'm probably not going to go into another career that's different from this one, because I spent a long time getting to this career and have a very specific skill set for it. But all of those skills are transferable, right? So from a bachelor's degree to a master's degree to a PhD and beyond, you have transferable skills in problem solving, in data analysis, in communication, and anything like that. Now, name a job in the world that doesn't want any of those skills, right? So ultimately, you could go almost anywhere. But more specifically in science and lecturing, I could end up doing more roles at the university, shaping the university, or I could get involved in more research, bringing in more money and bigger projects. The world is my oyster. I can't give you an answer to that one because, again, I feel like I've got the big boy job now, and I don't know what my career is other than this. Well, it doesn't feel like a job. I just enjoy doing it. Would you be able to use bioelectricity to look into synesthesia? Oh, yes is the short answer. How, I have no idea. I don't know much about synesthesia. It's way out of my realm. But yeah, it may well be interesting to look at a population of people that are synesthetic and whether they have different brain waves at different times of the day or responses to different signals. Yeah, I mean, bioelectricity is everywhere. So yeah, Someone's probably done it, but I couldn't answer that anymore because I've never done any research into synesthesia. I might have to after this because you've ignited my curiosity. I'll tick that one. How are we doing for time, Zach? Got time to fly through a few more?
Yeah, I think a couple more. I think two more, and we're all good.
Okay, perfect. I'll cherry pick two more then. These are anonymous, so I can't pick what I do or don't like. Um, did you plan on eventually? How did you get into career or research after degree in neuroscience? That was probably useful, so I'll go for that one. How did I get into a career of research after a degree in neuroscience? Um, I kind of just didn't stop doing it. I stayed in neuroscience with a master's degree, and then kind of stayed in neuroscience again with a PhD. Right? You don't necessarily need a master's degree or a PhD to do the research, because often you could work in industry where you could be in a research role without having, say, a PhD, for example. But often, if you want to be more involved in the leading of the research, then a PhD is particularly helpful for that. So getting into a research career really was me reaching out to um, uh, researchers that I was interested in their work and saying, can I come and join your lab for a bit and do something with you? So I recommend doing that. So that's that one. I will answer one more. I will have to answer that bottom one briefly, Zach, but it will be a one-word answer. Tell me you enjoyed praying more heart. Oh my word! Oh, they're all such great questions though. Um, which is helping with research different purposes and why. Yeah, I think that's a good one as well because a lot of you might be thinking about applying to medicine, and Brighton and beyond. Would you suggest that helping with research in university is a good thing for medical students to do, and why? Yes, I definitely would. I think it broadens your horizons, and gets you more aware of what's beyond the medical textbooks that you're looking at, and there is a lot beyond the textbooks. So I think that's really useful. I think as well, if you're looking to go on and become a consultant and a world-leading medic that is looking to cure diseases, realistically, they will also be researchers as well. So having some research experience at the earliest possible opportunity, it cannot be a bad thing, right? So yeah, I would actively encourage it as much as is possible, whilst obviously giving as much due diligence to your course and getting those grades to pass the course. So I hope that's my answer for that one. I have just seen this one at the bottom, and I will say, yes, it is a potential treatment for Alzheimer's disease, but it's a potential one, right? I mean, research moves slowly, and it'll be a long way until we know if we can properly use it. My research said with ApoE4, it's potentially interesting. So ask me again in 10 years' time, and I might be able to tell you. Cool. I could answer these all night, but I think Zach is asking me to stop, so I'll pause.
Yes, we'll finish it up there. There's loads of really good questions. With my sort of admissions and outreachy sort of hat on, I've seen a lot of questions about applications and university courses and what it's like to study these things. My top tip for you is to go to those universities at open days. Talk to current students who are doing those courses. Talk to their, academic teams who are running them, and learn for yourself from that experience. That tends to be the best way to find out about teaching styles and whether a course is right for you. And a crucial one is that courses are very different at different universities. So what might be doesn't appeal to you at all at one university is going to be very, very different somewhere else. So just explore. If you've got interests, go and explore and do some research of your own. There you go. We've been learning around research all evening. You can do some research now as well. Thank you so much, Ollie, for your lecture this evening. It's been really, really interesting. Thank you all for your questions. Lovely. That is all that I've got to say. So once again, thank you so much to Dr. Steele for your lecture this evening. It's been really fascinating, and thank you all for coming along and giving up your time this evening as well.